Hello, everybody. Welcome back. So in our last lecture, we were sort of discussing the complicated relationship between genetic inheritance and the environment. And now in this lecture, we're going to take this even further and understand genetics and behavior. And one of the most important examples we have of this was detailed in your book, looking at the idea of stress and mental development. So we know that there are alleles of serotonin transport protein that specifically react with the environment that an animal was raised in. And in this example, they specifically looked at monkeys that had experienced early stress in their life and understanding how that reacted with alleles that are responsible for serotonin transport protein. And this is really an excellent example, an animal example, of understanding potentially clinical depression. So we know that serotonin is an important signaling molecule in the brain. And basically, if you have serotonin transporter, these levels can affect different cell signaling that can happen in this area of the brain. And this graphic just sort of shows precisely what that potential mechanism can look like. And this is obvious. This is still a large area of research, but there is enough evidence to suggest that there is this connection between serotonin alleles and a susceptibility to environmental stress. So let's take the example of looking at clinical depression in humans. So there's an important study that was outlined in your book where we looked at clinical depression diagnosed in participants that had 2 copies of the serotonin allele and had experienced 4 or more tumultuous events in their life. And we see that people with 2 copies of this short allele affect the percentage of people that were diagnosed with depression. So again, we're starting to see this complex interaction between the number of stressful life events experienced and the presence or absence of this specific allele. And the result is this complex behavior of clinical depression. Now, previously, we also started looking at different behaviors of animals that are passed down. So we had the example of birdsong from previous lectures. And again, that's the understanding this complicated relationship between the environment in which a bird was raised, whether or not it had access to singing parents, and also their specific genetic makeup. So again, starting to unravel this complex interaction between environment, genes, and gene expression. Another important area that we haven't really talked about is this idea of potential genetic error. So we've previously sort of been looking at natural gene expression or influence or specific diseases that are influenced by specific gene structures. But we haven't actually really covered what potential errors in meiosis might have and how those generate specific genotypes and phenotypes. So the first one that we're going to be talking about is this idea of non-disjunction, and that's basically the failure of chromosomes to separate properly during cell division. And the second is this idea of aneuploidy, which is basically an abnormal number of 1 or more chromosomes. And possibly the most famous example of aneuploidy is this idea of trisomy 21 or Down syndrome. And your book does a good job of understanding potential risk with maternal age because, again, the number of eggs that a woman has is essentially that number that they have for life. And as those age, there's a potential for more errors to happen than eggs that are younger. So let's look at visually what we're talking about when we're discussing this idea of non-disjunction in meiosis. So for example, here we can see non-disjunction in meiosis 1, so where we have this original split and we see that non-disjunction occurs and we have more genetic material in this cell than we do in this cell. And this is the result of these gametes having too many chromosomes compared to when when normal meiosis takes place and no disjunction occurs, you have the same number of chromosomes that you originally had. Similarly, you can have non-disjunction occur in a couple different places. Here we have it happening early on in meiosis 1, but this can also happen in meiosis 2, and you can see that some gametes will have too few chromosomes and some gametes will have too many. And you can see that there's differential differences between each of these daughter cells. So since we know that these errors in meiosis can occur, how do we actually start to detect potential errors? And the main tool that we have at our disposal is to look at the karyotype of individuals, and in order to do that, we need to extract fetal DNA. And so the easiest way to do this is to perform a procedure called amniocentesis. And that's essentially where we're extracting cells from the liquid in the uterus, and in the womb, I apologize. And you can see, like, when we actually do this karyotype, so basically we're trying to get all of the cells in typically metaphase stage where we have those really condensed chromosomes, and we line them up, and we see here trisomy 21, where we have an extra copy of chromosome 21, which would result in the phenotype of Down syndrome. So I know we had kind of a lot of terminology and different types of gene expression that were sort of thrown at you for this section, but let's go through and really sort of capture what we've gone through. So we know that humans have 23 pairs of chromosomes, and one of those pairs is the biological sex chromosomes. But we also learned that sex determination has lots of genetic, hormonal, anatomical, as well as behavioral aspects. So it's much more complicated than whether or not a person has an X or a Y chromosome. We also looked at understanding how the Y chromosome can be used to establish paternity and why we typically use the Y chromosome to do these types of lineage checks. We also examined specific sex-linked disorders that can happen with traits that are inherited on X chromosomes. Again, these are called X-linked traits. And we know that they're more common in males than females because, again, the female has 2 X chromosomes, so there's the opportunity for one of those X chromosomes to mask potential problems in the other X chromosome. We also looked at a variety of different types of dominance. We looked at incomplete dominance, where we have that intermediate phenotype between the homozygous dominant and the homozygous recessive. We also looked at codominant traits, understanding this idea that both maternal and paternal alleles contribute equally as well as separately to a phenotype. And we also looked at polygenic traits, where we know that traits are influenced by multiple genes, and that these typically traits show sort of a continuous or bell-shaped population in the distribution. And lastly, we really examined this idea of understanding that phenotype can be determined both by genes and environmental influence. And this type of inheritance is multifactorial. We also looked at some specific differences in genetic disorders that can result from errors in meiosis. So where we have chromosome numbers that differ from the usual 46. And from all that, we're really starting to get a picture and an understanding of how the code in the genetic code that from DNA, how that gets expressed into a protein, and then how then those genes come together to create a physical trait or a phenotypic trait. So that's all for these lectures. We're nearing the end. We have about one more week of going through all this, and then we have our final exam. So we are well on our way to completing the class. And please, if you haven't taken the course survey, please do so as soon as possible. I really do take the advice and those reviews very seriously, and I appreciate your cooperation in that matter. Okay, thank you so much, and I'll see you next time.